discuss the topic clusters and tandem repeats and microsatellite repeat sequences. The microsatellite, it is a repeated DNA sequences in which the certain DNA sequence, which may be 1 to 6 or more base pair, are repeated in a DNA stretch, and the repetition may be up from 5 to 50 times. The microsatellite occurs at thousands of the locations within an organism genome, and they have a higher mutation rate than other areas of the DNA, and it leading to the high genetic diversity. The microsatellites are often referred to as the short tandem repeat sequences and represent as the STR by the forensic geneticist and in the genetic genealogy, or as a simple repeat sequences by the plant geneticist. Microsatellites and their longer cousins, the mini satellites, together are classified as the BNTR, means variable number of tandem repeat DNA sequences. The name satellite DNA refer to the early observations that centrifugations of the genomic DNA in a test tubes separate the prominent layer of the bulky DNA from the accompanying satellite layer of the repeated DNA. They are widely used for the DNA profiling in the cancer diagnosis, in kinship analysis, and in the forensic identification. They are also used in the genetic linkage analysis to locate the gene or mutations which are responsible for the given trait of the disease. The microsatellites are also used in the population genetics to measure the level of the relatedness between the subspecies, group, and individuals. Now the structure, locations, and the functions. As in the picture, you can see it is a or microsatellite. It is a tract of the tandemly repeat DNA motifs that range in the length from 1 to 6 or up to 10 nucleotides and are typically repeated 5 to 50 times. For example, the sequence Ta means Tata is a dinucleotide microsatellite, and another sequence is the GTC. It is a trinucleotide, and microsatellite which have the adenine, guanine, cytosine, and thymine. Now the repeating units of the 4 and 5 nucleotides are referred to as the tetra and the pentanucleotide motifs respectively. The most eukaryotes have the microsatellites with the notable exceptions of the some yeast species. The microsatellites are distributed throughout the genomes, and the human genomes contains around 50,000 to 1 lakh dinucleotides microsatellite, and lesser number of the tri, tetra, and the pentanucleotide microsatellites. Many are located in the non-coding parts of the human genomes and therefore do not produce the proteins. The microsatellites in non-coding regions may not have any specific functions and therefore might not be selected again. This allows them to accumulate the mutations unhindered over the generations and gives rise to the variability which can be used for the DNA fingerprinting and for the identity purpose. Other microsatellites are located in the regulatory flanking of the intronic regions of the genes or directly in codons of the genes. Microsatellite mutations in such cases can lead to phenotypic changes and diseases, notably in triplet expansions. Diseases such as the Fragile X syndromes and Huntington diseases. Now the mutations mechanism and the mutation rate in microsatellite. Unlike the point mutations, which affect only a single nucleotide, The microsatellite mutations lead to the gain or loss of an entire repeating unit, and sometimes two or more repeating simultaneously. Thus, the mutation rate at microsatellite loci is expected to differ from the other mutations rates, such as the base substitution rates. The actual cause of the mutations in the microsatellite is debated matter. One proposed cause of the such length changes is the replication slippage, which caused by the mismatch between the DNA strands while being replicated during the meiosis. DNA polymerase, the enzyme responsible for the reading DNA during replications, can slip while moving along the template, strand and continue at the wrong nucleotides. DNA polymerase slippage is more likely to occur when a repeated sequences. Because microsatellite consists of the such repeated sequences and the DNA strands may occur or make errors at a higher rate in these sequence regions. Now the several other studies showed that slippage is the cause of the microsatellite mount mutations, and typically slippage in each microsatellite occurs about once per thousand generations. The slippage changes in the repeated DNA are the three order of the magnitude more common than the point mutations in other parts of the genomes. Most slippage results in the change of the just one repeated unit, and slippage rates vary from the different allele length and repeated unit size. If there is a large size difference between the individual alleles, then they increase the instability during the recombinations of the meiosis. Now the microsatellite mutation rate. Microsatellite mutation rate vary with the base positions relative to the microsatellite and repeat type and the base identity. Mutation rate rise specifically with repeat number peaking around 6 to 8 repeat and then decreasing again. Increased heterozygosity in a populations will also increase the microsatellite mutation rates, especially when there is a large length difference between the alleles. This is likely due to homologous chromosomes. Now the biological effects of the microsatellites mutations. Many microsatellites are located in the non-coding DNA and are biologically silent. Others are located in the regulatory or even coding DNA. Microsatellite mutation in such cases can lead to phenotypic changes and diseases. A genome by the study estimates that microsatellite variations contribute around the 10 to 15% of the heritable gene expressions. Now the effects on the proteins. In mammals, 20% to 40% protein containing the repeat sequences of the amino acid coded by the short sequence repeats. Most of the short sequence repeats within the protein coding portions of the genomes have a repeating unit of 3 nucleotides. Since that length will not cause the frame shift when mutating. Each trinucleotide repeating sequence is transcribed into a repeating series of the same amino acid. In the yeast, the most common repeating amino acids are the glutamine acid, asparagine, aspartic, and the serine amino acid. Length changes of microsatellite in the effect on the gene regulation within the promoters and other cis regulatory regions can change the gene expression. The human genomes contain the many short sequence repeats in the regulatory regions, which provide the tuning knobs of the expressions. Now effects with the intron. Microsatellite within introns also influence the phenotypic through means that are not currently understood. For example, a GAA triplet expansions codon in the first introns of the H25 gene appears to interfere with the transcriptions and cause the Friedreich's ataxia. The tandem repeat in the first introns of the asparagine synthetase genes are linked to acute. A repeat polymorphism in the fourth introns of the NSO3 linked to hypertension in a Tunisian population. Now effect with the transposons. Almost 50% of the human genome is the contained in the various types of the transposable elements, and many of them containing the repeated DNA. It is probably that the short sequence repeated in those locations are also involved in the regulations of. Now in case of the cancer diagnosis, in tumor cells whose control and their applications are damaged, microsatellites may be gained or the loss during the mitosis stage. Hence a tumor cell and the line might show a different genetic fingerprints from that of the host tissues, and microsatellites have therefore been routinely used in cancer diagnosis. The marker assisted in the detections of the plant breedings and selections of marker aided selections. It is an indirect selections process where a trait of interest is selected based on a marker linked to the trait of the interest, and rather than on the trait itself. Microsatellites have been proposed to be used as a such markers to assist the plant breedings.